Okay, so I'm here with Lurong Pan, the CEO of Ainnocence. And so today we have some popular questions asked by many users. The first question is, what is the definition of artificial intelligence, also known as AI?
Good question. So there are actually two answers. One is the textbook answer, and another is, maybe everybody can understand type of answer. So the textbook answer is AI is an intelligent agency or agent. You give him a goal, it will optimize itself and perceive the environment and achieve the goal by itself. Which sounds kind of abstract. So, but there's another more general definition of AI is a artificially designed machine or other type of agents that can think, act, perceive as a human being or even beyond human intelligence. I hope that answers your question.
It does, thank you. Okay, so the next question is, how can AI help the rate limiting steps in drug discovery? And so basically, I think the way to break this down is as follows. What are maybe the top two rate limiting steps in drug discovery? And then the second part would be then how can AI help to accelerate those steps?
So I think that generally, it depends on how we view the drug discovery process, but I think generally there are two rate limiting steps. One is to synthesize or make the candidate compounds or candidate biologic molecules. Another rate limiting step is testing them in various stages, in vitro and in vivo. So in terms of those two steps, I think AI could help in two ways. First is trying to shortlist what need to be synthesized to be produced for testing. So in the conventional sense of setting that drug discovery process, the first step is screening all possible candidates. And normally in a conventional big pharma setting, you need to do you need to do a high throughput screening, which would up to like hundreds of thousands at least, or not, if not millions of candidates. And if for biologic screening, it could be like tens over 8 of magnitude different like biologically synthesized sequences. So in that case, it normally would spend at least as much of those like wet lab material to actually perform those screening. And the hit rates normally are really low. It's almost like 1 out of 10,000. In this case, that's really a waste of resource and time. And AI can help is shortlisting that to up to about 100 molecule to reach the similar activity bar that a project requires. So that's the first thing I think AI can help is really reduce the number of synthesis and the number of tests to be done in the primary screening at least. Another thing is we need to go through many different stages of tests from in vitro to in vivo, from animal to human. And in that case, we're actually testing those candidates in different biologic system of various degree of complexity. In that case, we will really need to make sure what we think in a simple system, such as a biochemical system, would really translational to a cell-based system or to an organ-based system, even to an in vivo complete system. So in that case, human mind is not able to really judge the molecule's property because we can only learn things from a very simple system, such as a chemical system. And it's really hard to really apply those rules directly to a biological events. In that case, AI could score all possible properties of a given candidate from its in vitro to its in vivo properties, and be able to really increase the chance of any chosen candidates to be able to successfully surpass all those test it way ahead and to reach the clinical or even to pass the clinical with a higher success rate. So in that case, is we do less experiment and with a higher chance of being successful, in short.
The next question is, Ainnocence has its own platform. And so the question that we get quite commonly is, how is that model trained?
Good question. So first of all, we in machine learning, general speaking, there is very industry recognized standard way and to do data collection, data assessment, and also the actual algorithm implementation to train those data. And also when the model is formed and how we evaluate those models, there's also have a standard process. I can give maybe a very short introduction to describe the process, but might not go into too much detail. So first of all, we have collected almost up to billions of different type of data points, which involves the information of molecule structure and its measured bioactivity, and also it's depends on the problem you're trying to predict. It's relevant data that has been collected in the previous experiment records that could be from patents or from other public publications, or it could be from a particular lab that donate those data. And after that, we would spend about 80% of the entire modeling effort on cleaning and sorting out those data to make it relevant and also reliable to the problems we're trying to answer. And then we're going to design a algorithm that be able to either perform a supervised or unsupervised learning task. So the supervised learning task is, we know we use the existing measurement or the label that we have generated in the previous test, and then try to train a model to be able to answer the same question. And unsupervised learning is almost like let the AI sort out those data and find the hidden pattern by itself. Such as in those cases, we normally do is like trying to just categorize different type of molecule into different chemical space, or trying to find out the hidden patterns of a different type of experiment measurement. So in that case, we first need to define that, and then we implement the algorithm. So the algorithm really nowadays has been really in a booming stage, so that we have the traditional machine learning algorithm that people would slightly put some of the domain knowledge to do, we call the feature engineering, and then we do approximation on those features we think is relevant. And another is deep learning, so that we would take more abstract input, such as just the string of the protein sequence, or just take the a string of a small string of describing a compound, and then we directly feed into a neural network of any kind, and then we approximate it to those labels. So, and that process take a engineering to really figure out the best black box that would giving the best model accuracy. And the model accuracy, normally we would have a series of matrix to evaluate them. But but in general speaking, there are two ways. So one is we call the prospective validation, and another we call this retrospective validation. So for the retrospective validation is normally the first step, because we have all those data in the history. We can split them artificially to mimic a testing and validation and training scenario. So in that case, normally we would do is we would artificially split those data by chronological order, such as if we have 20 years worth of data, we take the first 15 years as a training set and the last 5 years as a blind test as a testing set. And then after we train the model, we apply to this latest 5 years of data and see if our prediction is aligned with the actual experiment test. And another way is we just do it random splitting. We split all the 20 years worth of data in a evenly distributed statistic distribution of chemical space. And then we 80% of them are training and 20% of them we just do as a blind test and see if this model apply to different scenario. And to do it more thoroughly, we normally do like X-fold of tests, like we split it randomly, like 5 times or 10 times to build 5 or 10 models. And then we apply the same process and do an average and also monitor the variability between the models and see if we are fully digest the information from the training set. And also let's see how the model performed. And the matrix normally would like there's several common matrix to evaluate the model can be applied to different scenario. There's AUROC, AUPR, and there's precision, there's record, and there's F1 score, and there's confusion matrix. Those might be confused to like people who are not in the statistical field, but those are kind of common tests. But in that case, we sometimes use a very special matrix, such as enrichment factor in a like top 1% of the population or top 2% of the predicted population, trying to say if we try to enrich like for example millions to like 10,000 and if we could how much better than a random guess. So that's another type of special matrix commonly used in type of virtual screening type of model. So those are what we have to do for the retrospective test. And then for the prospective test, that's actually more easy to understand, is we been given a task of a screening project, and then we just use our model to really design the molecule and shortlist into like 10 or 20 or 100 compounds or antibodies, and then we go ahead test in the wet lab, and then let's see like how much that's actually has a better or a qualified experiment measurement, and the percentage of that we call it hit rate. So normally if when we practice this exercise, a prospective hit rate actually more than 1% would be already much better than the conventional blind high throughput screening. So the bar is not that high, but we're actually trying to shooting for 20-30%. So in that case, like Ainnocence has been trying to accelerate those models in the past experience that will really be able to hit as much higher as 40% in our best scenario, and with an average of like at least 10% of hit rate. So I think that's already an increase of industry bar. I think the final test of how different of our AI platform as opposed to other AI platform is the actual test in the prospective validation process. In terms of other properties in the qualities we assess AI is, we normally would also, besides its application, capability on the actual biologic problem, we would have other engineering perspective assessing an AI model, such as its computational efficiency, and such as its ease of use, and also its computational infrastructure that related to those fields. So in that case, our computational efficiency compared to conventional docking is actually 10,000 times more efficient, because we completely bypass that geometric description of and biophysics of scoring function computation process. We're basically using a pure data-driven deep learning strategy to achieve a better accuracy and at the same time much better efficiency. So we don't really need a super large computational resource to perform an even near cell complexity system. So this is the first engineering perspective. And another one is we deployed all our models and a service on a secured cloud on the Amazon cloud space. So in that case, we actually could eliminate a lot of software that you have to install and maintain and locally, and also the security is actually governed by the best cloud service in the world, the Amazon cloud. So and also it's in different continents. So engineering perspective, we will be able to serve different clients and different geological location and complying with their local data and security regulation, and which leverage the capability and infrastructure Amazon already established. So, so for short, we have a pretty validated AI system and being tested in multiple biological cases, and at the same time, we are really state of the art in terms of AI engineering. Thank you.
Great. And then I think you began to touch on this, but can you just briefly describe how Ainnocence platform is different from other platforms?
Yeah, sure. So I think in general speaking, there are 3 generations of platforms that you can see in the market right now. When I say this generation, it means the time being that they have been invented. So the first generation it probably invented about 40 years ago is purely biophysics-based underlying algorithm, which means you calculate the interactions between atoms. Normally, it's a classic mechanics approximation of a quantum mechanics, because we can't really directly apply quantum mechanics to a computation as large as a protein system. And in that ways, people have developed all kind of force field and trying to use empirical data to calibrate those force field as well, and trying to mimic a real-time simulation of the behavior of molecules and their interactions. So this approach in short, there's several terminologies people apply those underlying algorithm to build applications such as we call the docking. Somewhere we calculate the free energy between 2 molecules, and in the water phase that we call the free energy perturbation, FEP methods that are commonly used in small molecule drug discovery. However, this first generation of computation is really hard to scale up to protein-protein interaction, and also it's really hard to simulate protein with high flexibility. So in that case, people are trying to apply more empirical data or statistic measurements to really surpass that that obstacle. So that there comes the second generation. So basically, based on certain docking or protein-protein 3D configuration, people apply certain machine learning data and to trying to improve the scoring function of docking. And then also to expedite the scoring process of docking. So we call the second generation, it's more like a deep learning and physics hybrid model. And this model works actually much better than the first generation. The scoring accuracy is increased, but at the same time, it's also still restricted to a 3D information to be available, and also that 3D information still has to be accurate, and also the protein itself cannot be very flexible, and at the same time the protein-protein interaction problem still cannot be solved. So in that case, Ainnocence was based on what happens in the history, we're trying to solve those 2 problems, try to really scale up to all kind of protein system with flexible protein conformation or even without structure. At the same time, we really wanted to calculate protein-protein interaction in a very large scale to be able to screen biologics more efficiently. So in that case, we invented the 3rd generation, which is purely deep learning. Then in that case, we completely eliminate all the inaccurate classical mechanic process in the whole computation. And we do not simulate the process, which means the molecular dynamic process of what's going on between molecules. We just basically predict the probability of any given molecule binds to another one, or the probability of having certain chemical, physical, or biological property of any given molecule, just by giving its sequence information. So there's a principle in deep learning or machine learning is when we try to train a model, we would like the input data to have the list but sufficient enough information redundancy. So in that case, we need to give the most simple input to represent the entire information space of that object we try to describe. And then the model itself would learn all the hidden information that in the latent space, which means in the black box, and be able to give an accurate answer. So in that case, we really simplify the input to a very large degree. We just basically for protein-protein interaction, we just take the input as two sequence, the antigen antibody sequence, and be able to predict its binding affinity, relative binding affinity among a group of them. So in this case, it's really out of time limiting step in computation process, which means structure generation and also 3D like molecule docking. So all those steps can be completely eliminated. We just take the sequence and then we predict the result. And that sequence will be directly linked to the experiment measurements of the sequencing machine, or what we've been sent for order to produce that molecule by other vendors, and then we take the molecule, then we do the test. So it will be really naturally connected to the wet lab experiment without any other type of redundant computation. And eventually we would like to build our platform as complete like wet lab and the computation we called the dry and wet automation complete. So in that case, we can generate the wet lab data and feed in the AI model, and then to train it better, and then go feedback to do more experiments, and then to eventually form a reinforcement learning cycle. That in that case, every reinforcement learning cycle, we only need a few data points to be able to increase a base model trained by millions of data by at least 5 to 10%, which is a very high efficiency use of data points. So in short, I think we have very different base model algorithm, which will only take the sequence information. And also we applied the reinforcement learning cycle along with experiment prospective validation process. Hope that answers the question clearly. Thank you.
And then just last question. So if one were to want to work with Ainnocence, what would the information, what information would be needed to be given to you in order to get the results?
Perfect. Let me take example for both small and large molecule. Sure. So for small molecule, actually there are a simple version, or there's a more thorough version of what's required. For the simple version is we just need the name of the target. Yeah, we don't even need structure or any other information unless you have like a very particular mutation on the target you're working on. We just need the name, and then we need what type of pharmacology endpoint you would like that your small molecule have, such as what potency you would like against this target. And then what other like ADME toxicity criteria you require this molecule to have in order for for it to be successfully reach the clinical stage. So basically, you just give us those two information, it would be sufficient to get it started. And since we will have all our scientific intelligence engine to figure out, how to inhibit or activate this targets, or how to, and we have all the AI model to really generate possible candidate molecules and also screen them, shortlist them, and send them for the test for you. So, so that's the short version. But another one is, for example, if you have a halfway project, you've already done some screening or you've already identified certain lead, you would really like to you stuck somewhere or you would like to have a better molecule, you give us all the preliminary data you have, and then we would put them in our AI system to retrain it, be more problem specific. And then leveraging your preliminary data to build a more accurate AI model specifically tailored for your project, and then deliver the same thing. So our delivery will be basically dozens of molecules for you to do the next stage of testing, and then we guaranteeing at least like a 10% of hit rate. So that's for the small molecule part. And for the large molecule part, in the case of what you would like to optimize, you can optimize, for example, affinity or humanization or developmentally at the same time, so that you tell us all those criteria you require for those bars, and then we would basically just using one or multiple model for you. And the second thing tell us is the sequence information of your antigen. It could be the entire antigen if you don't know the epitope, or it could be a very specific epitope of interests. So in that case, you could we just need those two information. We'll be able to either give you a de novo design of antibody to have those criteria, or if you already have certain starting point of certain antibody you have in mind, or you tested them before, but it need to be improved, and then you can give that that primary sequence, and then we can further improve it for you, and we can even find the epitope for you if the epitope is not known. So that's kind of the what we need from you and what we would deliver.
Perfect. Okay. Thanks so much. We will post the contact information for Ainnocence, and look forward to future sessions.
Okay.
All
Thank
right.
you.
Thanks so much.
Thank you.